My lab certainly studies multiple multi-subunit enzymes. I'm just going to tell you a short story about one of them today, a protein called MIF, macrophage migration inhibitory factor. This is a master immunoregulatory protein that also has 30 or more different moonlighting interactions throughout the cell. So it engages in many protein-protein interactions, the structural and molecular basis of which are really not understood at all. Its primary function is to facilitate inflammatory responses, particularly turning on inflammation. It's overexpressed under all conditions of inflammation. It's found in high levels at sites of injury and inflammation in disease. And so there's also a really strong biomedical driving force for understanding this system. If we consider all of the inflammatory pathways found in the cell, let me get a laser pointer here. If we consider all of the inflammatory pathways found in the cell, MIF is involved in each and every one of them. But even if we look at just one small slice of this cell, in particular, the activation of the CD74 receptor, which is widely believed to turn on multiple cascades to promote inflammation. Even within this small snapshot, MIF has 5 or 6 different published interactions that have been purported to exist. And so there's a lot of desire to understand the functional promiscuity of this protein. And what was really puzzling to us was how such a small system, only 12.5 kilodalton monomer, or a 3-fold symmetric homotrimer, could engage in 30 or 40 different potential interactions in the cell. And so we really wanted to solve this problem at the molecular level. An initial trip to the PDB provided us almost no help, because if you look at 100 crystal structures of MIF in the PDB, many of them in the presence of mutations, small molecules, binding partners of any kind, there's essentially no structural difference whatsoever between them. They all look almost exactly the same. And so I'm showing you just an overlay of 30 different structures here. And you can see that there are really no conformational cues to provide us with information about how different interactions might be facilitated. However, when we began studies of this protein with different angles in mind, we started collecting NMR data on the system, and we found that under many of the same conditions, probed by crystallography, the NMR spectra look totally different, right? Almost none of them ever overlap. And so what this tells us is that MIF samples a pretty wide structural ensemble in solution that you can't really detect via X-ray. And so what we think is that NMR is a really good tool to understand the function of this protein. However, one particular insight that we were able to glean from analyzing these crystal structures was that of the importance of a solvent channel residue, tyrosine 99, this tyrosine that kind of sticks out into solvent space in the 3-fold axis of the MIF trimer. We originally started exploring this residue because it had one of the only evidence of conformational variability, this kind of 1.4 angstrom fluctuation in all the crystal structures. But we were initially trying to design small molecules that would bind there to lock the trimer together, potentially to facilitate some sort of function. And when we started mutating this residue in an effort to understand how small molecules might bind, we found that depending on the identity of the mutation, as well as the adjacent histidine 62 residue, we could modulate the catalytic activity of MIF above or below that of the wild type. And one of the reasons we speculated that might exist was because of this sort of pathway that exists between the solvent channel tyrosine 99, this is sticking out into solvent space, and its proximity to this N-terminal proline 1, which is a very rare catalytic nucleophile that exists in MIF. Only 3 or 4 proteins have this exceptionally low pKa proline that catalyzes an enzymatic reaction. And so we then shifted focus to studying tyrosine 99 and its associated residues as an allosteric switch that might mediate catalytic function. And this is about 9 angstroms away from proline 1. Okay. Our first crystallographic exploration of this system led us to come up with a relationship between general flexibility of the MIF structure and its catalytic function. Okay, as I showed you on the previous slide, some mutations of tyrosine 99 and histidine 62 could modulate activity above wild type, and some would really severely attenuate activity. And it turns out that mutations that create a highly active species of MIF above that of wild type have aromatic or very bulky side chains as the mutation residue. And what they end up doing is creating this very ordered network of solvent molecules and hydrogen bonds in the solvent channel, such that it almost closes off this central solvent channel and really rigidifies that structure, okay? And this propagates somehow to the proline-1 active site, okay, the N-terminal active site. On the other hand, if we make alanine or glycine mutations, things that really introduce flexibility into those sites, we see almost no hydrogen bonding and no order of the solvent network, as well as this expanded solvent channel hole here in each one of these donuts, and they have severely attenuated activity. So what this is telling us initially is that there's a relationship between the rigidity or flexibility of this active site and the catalytic function that we observe. We explored this further. Again, I'll call you back to the initial plot of catalytic function, right? We can see that some activities are really, really low. Some are actually above that of wild type. And what we found with NMR studies of these exact same mutants was that glycine and alanine mutations introduced a huge amount of line broadening into the NMR spectrum, evidenced by these pink bars here in the per residue plots of the chemical shifts. And so what this again confirmed to us is that the more flexible the entirety of the protein structure becomes in this case, particularly the region around the solvent channel and the catalytic site, the more likely the enzyme was to have a very low activity. And when we introduce these bulky aromatic residues here, you could actually see in the crystal structures of the mutants that the side chains were becoming stabilized by additional stacking and weak interactions that were bringing them closer together, okay? And so again, there's this flexibility activity relationship that we explored, and I'll just leave it at that for now, but I'll happily talk more about that after the talk in a breakout room. Interestingly, while we were exploring NMR-related correlations via chemical shifts and dynamics, MD simulations also showed that tyrosine 99 was dynamically coupled to the N-terminal enzymatic site, as well as the C-terminus of the adjacent monomer, okay, and shown here in this kind of blue ribbon. And that was particularly interesting because the C-terminus is proposed to be the binding site for the CD74 receptor that turns on inflammatory action in the cell. Okay, and so MD simulations revealed that all of the colored sections of this structure were dynamically correlated to tyrosine 99 in the solution. So this encompasses all the way here, the N-terminal active site and the C-terminal CD74 binding site, which led us to propose that if we examine CD74 activity in the presence of mutations, it might be affected by tyrosine 99, and that's exactly what we saw. So in vivo, tyrosine 99 mutations actually turn off CD74 receptor activation, even though they have nothing to do with the binding site. And so this led us to believe that Y99 could be like a central hub of regulation in between these two sites that almost exist in a perfectly straight line. Okay, the enzymatic activity at the C-terminus and the CD74 receptor activation at the C-terminus. And what we observed is that the reduction in catalytic function, in this case for a tyrosine 99 to alanine mutation, was different in both of these cases. And we wanted to understand mechanistically how this was the case. One of the things that we turned to was looking at simple NMR chemical shift correlations to see if there was in fact some coupling between these sites. What I'm showing you here in this panel is that in the presence of tyrosine 99 mutations, tyrosine 99 indicated by this green sphere here, the MD simulation data that I just showed you in the previous slide is summarized, effectively saying that all of the spheres on this structure are residues that are correlated dynamically to tyrosine 99 in the MD simulations. That includes the adjacent C-terminus, and that includes the N-terminus that houses the enzymatic activity, as well as sites in between that. If we look at NMR chemical shifts of the same tyrosine variants, the picture's essentially recapitulated. So what we observe is that NMR chemical shifts are felt at the N-terminus and the C-terminus and surrounding areas, suggesting again that perturbation of tyrosine 99 is propagated in a way that is felt by all of these regions of the protein. However, interestingly, if we make the same mutation at proline-1, we do not see the reciprocal chemical shift changes at the C-terminus, only at tyrosine 99. And in a true allosteric system, this was puzzling to us because all of the different regions should be functionally and thermodynamically coupled to each other. So we were expecting to see chemical shift perturbations at this site, and we haven't really been sure what to make of that data yet, but we're working on different angles that I'm happy to talk about in the breakout room. Needless to say, we expanded on that initial paper. and studied the only other MIF homolog found in humans, D-dopachrome tautomerase, which is also a pro-inflammatory protein that overlaps in activity with MIF. It binds the same receptor. It has the same threefold symmetric trimer structure. However, it has only 35% sequence homology to that of MIF. And as it happens, with the exception of proline-1, none of the residues in these two enzymatic or CD74 binding sites are the same. Okay, and so we asked the question basically, would functional coupling be preserved between tyrosine 99, in this case phenylalanine 100 by the new sequence, in this enzyme that doesn't share any sequence conservation with MIF? And it turns out the answer is yes. So regardless of the residues that are present, if we make mutations at these residues here, the same exact ones that I've been talking about for MIF very briefly, we see that activity is modular and we can turn it off a little bit. And the same thing goes in vivo. We can knock out CD74 receptor activation, both with mutations at the N terminus, which is completely far away from the C terminal site, as well as the allosteric site. Okay, so this was exciting to us to suggest that potentially this was a conserved mechanism in the protein. And we wanted to explore this in MIF2 DDT, this new enzyme, using NMR as well, and try to make connections between the two proteins. And so what I'm showing you here is essentially a matrix of NMR chemical shift data. Along the top here are residues that correspond to sites within this pathway, if you will, these two sites, enzymatic and CD74 activation site. And along the y-axis here are the three mutations that we studied. And what you see in each panel is that there is a chemical shift effect or a line broadening effect caused by each and every one of these mutations on these different residues, suggesting that the effect of the mutation is felt along this pathway in each case. And this is summarized structurally on these slides here, or on these pictures here, where you see in green the location of each one of the mutations that we introduced. And in orange and blue spheres, you see all of the residues that respond via chemical shift effect to this mutation. Okay, and we can see that in each case, the adjacent C terminus is being activated, as well as the N terminal active site in most of these cases, to varying degrees. Again, generally the picture is that this is preserved. However, we need to work out a lot of the intricate details. We went a step further and used spin relaxation parameters to also probe the dynamic effect and correlation experimentally between these systems. And what we found is that most of the residues that have altered dynamic parameters from that of the wild type in each of these mutants correspond to sites within this allosteric pathway or that directly surround the enzymatic or CD74 sites. Again, suggesting that there is some level of coupling between these sites. And really, we just need a little bit of time to work out exactly what that is. And the last thing I'll tell you in this sort of brief talk is we've been trying to make a lot of sense of the CD74 activation data, particularly because it doesn't recapitulate the levels of enzymatic attenuation or enhancement that we observe. It kind of does its own thing. And CD74 has never been really characterized structurally in complex with MIF in any way. We've generated a soluble extracellular construct of the receptor that we can get modest NMR chemical shift perturbations out of. It turns out those perturbations localize around the trimer to areas that we would expect at the C terminus in between the interface of the monomers. However, we're working on optimizing those data for NMR. In the meantime, we've been predicting structures of the complex via docking and simulation, looking at energetics of different orientations of the CD74 receptor on MIF, and using community network analysis via MD simulations to try to understand how crosstalk between different regions of the MIF protein, which we've characterized in the previous slides, is rewired in the presence of CD74. And more simply, we've been looking at general dynamic fluctuations of the MIF structure across the entirety of the trimer over MD simulations and looking at changes in allosteric sites and active sites. And what we found is that in the presence of CD74, so far, the allosteric region, this kind of central solvent channel, becomes more flexible, while the C terminus, the binding site, becomes a little bit more rigid. This makes sense because biochemical studies show that CD74 binding actually almost completely attenuates catalytic function, and this would be consistent with the idea that this is becoming more flexible and therefore attenuating activity, which we observed in the previous mutational analysis. I hope that wasn't too much information in a very short time, but as I said, in the breakout rooms, I can provide more information. We have a really cool avenue that we're also exploring in a different way to look at allosteria and MIF, but I just want to thank the lab for their work on this project, particularly Emily Chen and Aaron Skeens, two grad students who really pushed a lot of this work forward. And of course, thank you very much for your attention.
Great. Thank you very much, George. We'll keep the question until the breakout rooms. So our next speaker is Siavash. Siavash?
Yep, this is correct. Yep, Siavash. Yeah, let me just share my screen and then I'll get started. Right. Can everybody see my screen okay?
Yes.
Perfect, okay. Let me just get this. Beautiful. You can also see my video feed.
Video feed?
Yep.
Oh, yes.
You can see me speak, yep.
Yes.
Perfect, okay, let's get started. First of all, thank you very much for the opportunity to speak here. Today, I'm going to talk about some of the work I did. My last work as a postdoc in the K lab studying the allosteric regulation of this lovely protease, ClpP1P2 from tuberculosis. So we used a bunch of different methodologies, including methyltrose NMR and cryo-EM. So first things first, let's talk about TB a little bit. So prior to COVID, TB was the leading cause of death from a single infectious killer in the world. Almost 10 million people became sick with TB, and over 1.5 million of those died. TB is the top killer of people with HIV, and it typically mostly affects people in countries that are developing with relatively poor healthcare infrastructure. So although the total number of cases is going down, the number of antimicrobial resistant cases is on the rise. And this is a cause for concern. And this calls for TB to become and be a top priority for research and development of new antibiotics. A little bit of a fact here is that over the past 50 years, only a single new TB drug has been approved. So we need to do more work in that area. Now, turns out that these antimicrobial resistant tuberculosis cells are resistant to a lot of these frontline antibiotics that we have currently at our disposal. So for example, antibiotic A and B, even when applied together, you still end up with this population of persister cells shown here. But if you add this compound called ADAP, the structure of that is shown here, you can also kill even the persister population of these resistant TB cells. And with a fair bit of work, both structural and also genetic work, has been established that ADAP in tuberculosis really dysregulates the function of this protease called ClpP. So the combination of these small molecules that interact with TB and specifically target the ClpP protease, and the fact that ClpP is essential for the viability of TB means that it is a prime and attractive novel drug target. So a little bit more about ClpP, the ClpP system in bacteria, and also TB, lots of similarities. If you're familiar with the proteasome, this is going to sound somewhat familiar. So ClpP is a barrel-like proteolytic chamber. The schematic of that is shown here. It's a double donut sort of structure, 7 donuts on top, 7 donuts at the bottom, and it's sequestered catalytic sites within the barrel. It needs to work together with AAA plus unfoldases. In this case, it's ClpX. ClpX engages substrate molecules, hydrolyzes ATP, and in doing so, feeds the substrate into the ClpP chamber for degradation. This is typically what happens in a regulated sort of environment. A little bit about the structure of ClpP. We have structures of ClpP from a large number of systems. This is the one that we published a few years ago. This is from Steph. So this is the side view of the complex. This is the side view of the barrel. If you turn it upside down and look at it from the top down, this is the axial port that allows substrates to get into the core of the barrel and get proteolized. Typically, clip X, as I mentioned above, is charged with feeding substrates into clip P. And if you look at the structure of a single monomer, there are three main domains. The N-terminal domain, colored red here, which is responsible for forming these axial pores. The head domain, the green domain here, which forms the main body of the barrel, and the handle domain, which is going to be the highlight of the show today, mediates inter-ring contacts and also helps really align the catalytic triad. Clip P is a serine protease. It's got this serine, histidine, aspartic acid catalytic triad. So we're going to talk about this handle region a fair bit today. Now, a clip P in mycobacteria is somewhat unique in that it does not, basically it's encoded by two genes, clip P1, clip P2. Most organisms, including human, have a single clip P gene. So we went right ahead and expressed these in E. coli. You get a fair bit of lovely protein to work with, high yield, it's relatively well behaved. And if you run them on a sizing column, clip P1 and clip P2, they elute basically as heptamers, not a tetradecamer. You basically get a half clip P ring. And we know this because this co-elutes rather nicely with the Steph clip P that I talked about earlier, that is well characterized. And this mutation R171A in Steph clip P leads to the dissociation of the tetradecamer into a heptamer, and these basically all co-elute. Now, let's talk about the function of these two, and this is where this system becomes particularly interesting. We use these peptidase assays to probe for function. So for basically every other clip P, what you can do is you can add these tripeptides attached to an AMC group. Clip P comes in, cleaves the bond here. This AMC group is released, its quantum efficiency, quantum yield goes up, and you get an increase in fluorescence that you can easily read off with a plate reader. Now, what's interesting is that in the case of clip P1 and clip P2 in tuberculosis, you basically get no activity whatsoever when in isolation. So clip P1, clip P2 on their own in isolation are dead. And ADAP, this regulator that I mentioned, also doesn't activate them when in isolation. Given that we have both of these now in high quantity, we can just mix the two of them, P1 and P2. You get a shift in the sizing column profiles, in the infiltration profiles, that now co-elutes with the Steph clip P. So it turns out that the two of them bind. One can relatively easily probe this and quantify these interactions using methyltrypsin NMR. So in this case, I'm looking at the isoleucine region of ILVM methyl labeled clip P1. That is the green spectrum, and the purple spectrum is when we have added an excess of uniformly deuterated and therefore NMR invisible clip P2. You see these characteristic shifts as a result of the addition of excess clip P2, indication of binding. We have full assignments for ILVM groups that I obtained, by the way, via mutagenesis. This is the methionine region. We also see these characteristic shifts. And we can do this as a form of a titration. And in that scenario, we get two sets of peaks. Some of them belong into the holo state, the bound state. Some of them belong into the free state that goes down in intensity. And we can fit them to a simple model, and we get a KD of 3.5 micromolar. So now we can easily form the complex. We know how to form it. We know how much to add. We can then mix the two. So clip P1 and clip P2, even when mixed, it's still catalytically dead. ADAP doesn't activate them. But what is known in the field is that if you add this bezel molecule, it's basically a dileucine with an end-capping benzoyl group, you get this activation in the, you get activity back in clip P1, P2. And this is really unique among bacterial clips and all the clips that I know of. And it turns out that if you have bezel and also add ADAP on top of it, this dysregulator, you get additional activation. So what we do not know, what's so special, unique really about the mycobacterial P1, P2 system is that we do not understand the lack of activity. We do not have any mechanistic understanding of why the apo form, even P1 in isolation, P2 in isolation, or mixed. At that point, we did not know why there's a lack of activity. We also didn't know how these short peptides, these dipeptides, how they activate the complex, the P1, P2 complex. So hopefully by the end of this brief talk, you'll have some idea about both of these. So we figured probably as a first step to try to understand why ApoP1, P2 is inactive is to obtain a high resolution structure. A structure of the bezel-bound form of P1, P2 was already available from the lab of our collaborator, Alfred Goldberg, and it shows basically the typical ClP structure that are available from a large number of bacterial systems. But the exception of the fact that bezel is basically bound at all 14 catalytic sites. So all 14 catalytic sites are fully occupied. But the structure of the apo form was not available. This was obtained using cryo-EM. So this sort of overall view shows you that the height of the complex, this is the side view of the barrel, is a little bit shorter, 83 angstrom relative to 87 for the bezel-bound form. And if I zoom out a little bit from the overall structure and I focus on opposite protomers in the context of the overall complex, you'll see that the handled region, which in the bezel-bound form and all of these other active bacterial clips is nicely formed. You have a nice helix here, and preceded by a beta strand that comes together and forms a beta sheath with the opposite protomer, and helps this whole handle motif really helps align and stabilize the catalytic residues, is quite disordered in the apo form. So in particular, there's a histidine residue that really helps align the catalytic triad that is missing. So if I were able to show this schematically, so this is the bezel-bound, this is the apo form where the handle region is quite disordered. The histidine residue is far away from the catalytic serine residue, and therefore we basically have no activity. But what happens when we add bezel is that bezel, despite the fact that it's only a dipeptide, it forms these backbone-mediated hydrogen bonds with the handle beta strands that are on both ClP1 and P2, stabilizes them, forms a nice sheet, brings serine and histidine in close proximity, and you get activity back. The sort of dynamic nature of the handle region is also evident in the cryo-EM local resolution maps. So the overall structure is of very high resolution, but then the handled region is really sort of sticking out as a bit of a sore thumb, indicating that this region is really floppy. Now, we've answered one question. We now understand why the apo form is inactive, but really the most interesting part for us was why this particular system requires these short dipeptide activators, and what's the mechanism of activation. So for that, we made heavy use of NMR spectroscopy. Given that we have full assignments of P1, we did the titration involving the addition of fully deuterated bezel into the spectrum. So we went up to 8 millimolar. And I'm showing only 3 residues here. But what you see, even with these millimolar type concentrations, we see the disappearance of one peak and the appearance of the other, which get modulated in intensity with additional bezel. This is for isoleucine 60, isoleucine 165, and methionine 75. We can also reverse the labeling pattern. Now P2 is ILBM labeled, P1 is uniformly deuterated, and PC is similar pattern. Now, if we follow these intensities, some of these go down in intensity, these peaks, some of them go up in intensity. This is the way they look like. This is for the P1 ring. This is for the P2 ring. This is your typical NMR titration. And if you look at the shape of these points, they do not correspond to the typical hyperbolic binding curves that you're used to seeing. These are highly, highly sigmoidal, which as I'm going to explain in a few slides, is going to be speaking about allostery. We can also do some measurements in terms of activity also as a function of Beazle. So if we look at the activity of the complex at a fixed substrate concentration of a millimolar, you see also that initially we have no activity, as I mentioned, but then we have this really sigmoidal rise in activity, and then it tapers off a little bit at the end. So these sort of sigmoidal curves are indication of allostery, which traditionally has been measured using the quantified using the Hill function. So if you fit the NMR data, for example, to a Hill function, we get a Hill coefficient of 7, which is highly, highly cooperative. So we thought this is really quite fascinating. And to really get a better handle on how the allostery properties of the system modulates its function, we figured we should come up with a model that explains all sorts of functional and structural aspects of activation. So we came up with a customized MWC model for the ClpP1, ClpP2 tetradecamer. So we have now a conformational equilibrium. So we have the sort of short compressed state of the complex P1, P2. We also have the extended taller one, which is active. We know this from our high resolution structures, cryo-EM, and X-ray crystal structures that we have. And we can basically invoke the presence of two states. One, a T state corresponding to the short form, and an R state corresponding to the extended active form. For the ClpP1 ring, the R state can bind a Beazle molecule, the activator, with an affinity called KR1X. It can also bind a substrate molecule, PKMAMC. We're going to call that affinity KR1Y. The T state can also do the same thing, KT1X, for the binding of a Beazle activator, and it can also bind a substrate molecule, PKMAMC. So this is just for the P1 ring. We can also have the exact same equilibria for the P2 ring. We have a T state for the P2 ring, we have an R state, and each of those can bind a Beazle molecule and a substrate molecule. Now we have a fair bit of data, both NMR, remember that we have some peaks that go down in intensity, some of them go up in intensity for the P1 ring, for the P2 ring. We also have functional assays performed as a function of substrate concentration. I'm only really showing a fraction of the data here. And we can feed all of those to our model, and we can do a global fit. And what we tried all sorts of combinations of fitting parameters to get robust fits to the data. But no matter what parameters, what sets of, what fraction of the model you're using and trying to fit, we get themes. And these are sort of conserved no matter what sort of model we use. One of them is that a KT1X is equal to KT2X, which is almost 0. This means that the T state of both the P1 and the P2 ring are incapable of binding the activator. So this sort of came out of the models without us really driving it in any way. The KT1X is much smaller than KR1X, which means that the R state of the P1 ring has a much, much higher affinity compared to the T state of the P1 ring. So basically the R state has a lot of affinity for the allosteric activator. And the KR1X is much greater than KR2X. So the P1 ring has a higher affinity for a Beazle activator. And this other thing that comes out of these fits is that the R state fraction is less than 1,000th of 1%, which is the active fraction, the R state, if you recall, which explains the lack of activity of the P1, P2 complex in the absence of our allosteric effector. Now, we can use some of these fitter parameters to get additional insights into how the allostery of the system modulates its function. I'm going to only talk about a fraction of that, and that is, for example, we can predict the number of Beazle molecules bound to the P1 ring. So if you look at peak activity here, occurs at about 4 millimolar Beazle. If you come down and look at the green curve here, you'll see that about 4 or 5 Beazle molecules are bound. And recall that P1 at max activity has 4 or 5 Beazle molecules bound. P1 is a heptamer, which means that the majority of the activity of the complex at max activity is really coming from 2 or 3 free, bezel-free, but yet active P1 protomers. So basically, you have this equilibrium, the inactive state, the addition of bezel pushes it towards the active state. This is the P1 ring, this is the P2 ring. Bezel can bind to both rings. And we have this situation here where bezel occupies some sites, as the crystal structure suggests. But here with NMR and modeling, we can quantify and find out that, yes, the number of protomers among as part of a heptamer that are contributing to catalytic activity is really about 2 or 3 of them. There are a lot more insights that we gleaned from these MWC modeling that led to more experiments, more fun experiments that sadly do not fit within the 10 or 15 minutes that I have here. So if you'd like to know more, please join us in the breakout rooms. Just a little summary, we talked about the TB crisis and how CLIP is a really good drug target. And we mainly answered 2 questions here: understanding the lack of activity and understanding the activation mechanism of the P1, P2 complex. We use a variety of methods, NMR to follow Alice theory, cryo-EM, some functional assays, and some MWC modeling to better understand the system. So this was work done in the lab of Lewis K, where I did my postdoc. It was published right before the pandemic. Since then, I have started my own lab at the University of Guelph here in Canada. So if you'd like to know more about what my lab is doing, also I'm happy to chat about that in the breakout session. All the beautiful cryo-EM data that you saw was done in collaboration with John Rubenstein and his then graduate student, Zeb Ripstein. He's now a professor at the University of Manitoba here in Canada. And yeah, happy to chat more about all of these in the breakout room.
Thank you very much, Siavash. Our next speaker is Sarah.
Can you all see this fine? Oh, sorry, I should play it.
Yes.
Yes, okay, excellent. So I'm really excited to be here today as well. And I'd like to extend my thanks to the organizers. So today I'm going to give you a slightly different talk. I'm going to talk to you about some of the educational initiatives that I'm working on to really bring bio-NMR into the undergraduate curriculum. So I'll start by introducing my favorite biomolecule, which is RNA. And we've all probably heard more about RNA in the last year and a half than in the many years prior. So I'm going to keep my comments very brief. What I want you to know is that RNA molecules are really important regulators in essentially every aspect of biology. And their structures and mechanisms of actions really need to be studied to fully understand their regulatory potential. Now, even with the growing awareness of the importance of RNAs in biology, our structural knowledge of RNAs lags significantly behind that of proteins. So in this snapshot from the PDB, you can see that RNA containing structures account for only 3% of the total structures, and that proteins really dominate our structural knowledge. Now, my lab studies both non-coding regions of messenger RNAs and non-coding RNAs, truly non-coding RNAs, that are important in regulating gene expression. And I hope to come back for another seminar a different day to share some of our research projects. But today I'm going to highlight, like I said, some of the educational initiatives to engage undergraduate students in biomolecular NMR spectroscopy of RNAs. So let's start at the beginning. You have a sample and you've collected your first spectrum, and it's beautiful. But without chemical shift assignments, you can tell very little about your RNA biomolecule. Perhaps whether it's folded or unfolded, but true rich structural information is really lacking. Now, whether you work with proteins or nucleic acids, chemical shift is a fundamental indicator of molecular structure. And as we heard from our previous speakers, really is at the heart of all NMR studies. But as I'm sure we can all appreciate, making complete chemical shift assignments can be a really time-consuming step that's required for the subsequent analysis of biomolecular structure, dynamics, and ligand binding. So efforts to automate the assignment process with chemical shift prediction programs, which Bruce Johnson, who's one of my collaborators, among others, have been leading, has the potential to really greatly accelerate this early stage roadblock. Now, Bruce's program in particular is trained on data that's pulled from the BMRB. One major problem with this approach is that the RNA chemical shift database is not complete. There are many missing assignments, and the accuracy of the chemical shift prediction really depends on having a robust and complete chemical shift database. In recent years, there have been less than 30 RNA chemical shift depositions per year. Now, these numbers are likely to change for 2020 and 2021 as several labs have really focused on studying RNAs from SARS-CoV-2, but nonetheless, the trend is that on average, very few depositions are made per year. Now, another potential complication is that the chemical shift database must encompass really a wide variety of sequence and structural motifs for accurate predictions. So now that I've told you what the problem is, here is one way in which we are working towards a solution. Our goal is to really expand the chemical shift database for RNAs. With Bruce's help, we've identified RNA sequences that are missing from the current database. And students who are enrolled in a techniques and biophysics class, now this is a mixed undergraduate, graduate laboratory class, gain authentic research experience learning how to assign NMR spectra of RNAs while contributing to our broader chemical shift knowledge base by depositing their assignments in the BMRB. So as we begin data, we considered a 5 base pair helical stem element, okay, and that's sort of diagrammed here. Now, if we consider only canonical base pairs, that's G paired with C and A paired with U, there are 64 possible triplets of canonical base pairs, and we define that as a core sequence, okay? Now, I'll make sure to mention here that the nucleotide of interest that we're gonna really focus on the completeness of the chemical shift data is this central nucleotide on the 5' strand of the RNA. Now, the base pairs that flank the core sequence are variable. And here we examined instances where we either had purine or pyrimidine-purine base pairs at these positions. Because these positions are farther away from the sort of nucleotide of interest, we really only considered nucleotide type rather than nucleotide identity here. So this gives us 256 different combinations of RNA sequences to examine. Okay, so the identification of RNA sequences that were underrepresented in the BMRB was done by Bruce, using tools that his lab developed for training models for the prediction of RNA chemical shifts. He basically took NMR star files containing RNA chemical shift data that were downloaded from the BMRB. And these data were searched for all 256 5 residue sequences. Okay, and I'm showing you a small portion of the data set here. And I'm highlighting, again, this core sequence in these rainbow colored boxes. Okay, so again, we have, I'll mention here also the shading, the color coding. Each core sequence has 4 potential variable sequences, whether purine, pyrimidine, or pyrimidine-purine at the flanking positions. And so we really define each of these as a group. So a similar core sequence with a variable flanking sequence, all of these RNAs are in a group. So group 1, group 2, group 3, so on and so forth. Okay. Now we generated a table containing the number of measured chemical shifts for each of the aromatic protons and carbon atoms for the corresponding sequences. And again, I'm showing you just a small region of this, and I've highlighted all of the RNAs that have missing chemical shift data. Okay, and what I hope you can see is that some groups are let's find a group. Some groups are really well represented in the BMRB. So for example, this group has 3 of the 4 elements have missing chemical shift data, where this last group, all of the RNAs are really well represented. So we found 182 groups have at least 1 assignment for all of the aromatic protons and carbons, and about 74 were missing at least 1 assignment for aromatic proton and carbon chemical shift data. So based on this sort of analysis, we identified 4 groups of RNAs to focus on. These RNAs were among the most incomplete groups of RNAs, where 3 of the 4 members had missing chemical shift data. So from here, we identified 12 RNAs, 3 from each of these 4 different groups. All of the RNAs we studied were put into sort of a common template. This is a simple hairpin. We've got a GC clamp at the base and a GA-GA tetraloop to sort of lock the hairpin structure. And the sequence that we vary is here, this 5 nucleotide sequence, which we pull from these 4 different groups. Perhaps an easier way to visualize this is to see the 12 RNAs that we're studying. Again, these are organized by group, and their common core sequences are color coded here. Okay, and flanking sequences you can see are variable within the group. Okay, so because of limits on time and the switch to virtual instruction for the pandemic, researchers in my lab prepared and collected the data on these 12 RNAs and actually pre-assigned all of the data. We then removed a subset of assignments. So for RNA 24, we removed the assignments for residues 15 to 20. Pairs of students in the class were then given the partially assigned proton-proton nosy and proton-carbon HSQC data for a single RNA. Having this partial assignment really allowed the students to learn through doing the rules for assigning their data set. So they could really focus on seeing the trends rather than trying to memorize some abstract rules. So for example, they were really easy to connect nucleotide 1 to nucleotide 2, nucleotide 2 to 3, 3 to 4, and so on and so forth. Similarly, having the HSQC data was really helpful for teaching them the differences between, say, or being able to really quickly detect a C2H2 from a C8 or a C6 correlation. Okay, so once students familiarized themselves with the data and the software that they had to use, they could continue the walk from where the pre-assignments left off. And so again, we removed the assignments for this block of nucleotides, so they could start at residue 14, connect to 15, 15 to 16, 16 to 17, okay? Now, at the end of the class period, right, we had a completely assigned data set really for each of these 12 RNAs. We then used NMR-ViewJ to validate our assignments. And again, for simplicity, I'll show you a single spectrum, but we did do this analysis on all of the RNAs. So the assigned chemical shifts for specific atoms in the RNA are sort of represented here, or presented here in a summary format. And the closed and open circles represent predicted values for chemical shifts. Now, the closed circles are predictions that are based off of data that's in the BMRB currently, whereas the open circles are predictions that lack this sort of database training behind it. So essentially, all of these open circles really represent new missing data that we're adding to the BMRB. We extended the study into to include the assignment of immunoprotons, which inform on secondary structure, and continue to build on database and prediction tools that were reported earlier this year from Yi's lab. So with these 12 RNAs, we really made a huge effort in the assignment process. We assigned, 330 aromatic correlations, aromatic carbon proton correlations, and 108 immunoproton chemical shifts. We essentially assigned all of the aromatic to anomeric protons for every nucleotide in the RNA, and a good number of additional ribose proton assignments. So this is really a massive effort. Okay, so I hope that you all can appreciate this sort of authentic research experience that I've developed and implemented for our students. This gave students really great exposure to fundamental principles of NMR spectroscopy. It gave them hands-on experience with the analysis of really brand new NMR data. This is not data that's been passed down from year to year, new data, so it's still really exciting to look through. I didn't talk about this aspect, but we also taught students a little bit about RNA folding and thermodynamics. And students got to really hypothesize and think about trends in chemical shifts and how different sequence and structural features affected the chemical shifts of their RNAs. And again, we've added really valuable chemical shift data to the public domain. So as you know, our job as scientists doesn't end with performing experiments. We have to also communicate our findings. And so the students also really got, I think, really great experience in this aspect of research as well. So I already told you we assigned the chemical shifts, and we've deposited the assignments and the raw data all in the BMRB. So hopefully other people could be inspired to implement these types of experiments or lab modules in their classes. And rather than a traditional lab report, the students in the class wrote a draft manuscript describing their findings. We then compiled all of their collective findings, and we've submitted this manuscript for publication. And so these are all of the students that did this work. Everybody who's in red is an undergraduate student, again, enrolled in this course. Graduate students in the course are shown in blue, and the students from my lab and of course our collaborator are in black. So with that, I'd like to thank you all for your time, my lab for their efforts on this project, particularly Yaping and Anita, Cookie, who's now a first-year student at Yale, and Tracy, our technician. Funding from the NSF. This is supported by my career grant, and of course our collaborator, Bruce. Lastly, shameless plug, we are looking for talented graduate students and postdocs. So if you are interested in learning about some of our research projects, please reach out. I'd be happy to discuss. Thank you.